Hello, everyone. Thank you for joining us for episode 9. Before we begin the formal presentation, I'd like to take you through some of the features of the GoToWebinar system. If you have a problem, please use the question field in the control panel on the side of your screen, and we'll try and get you some assistance. We'd also like to encourage you to ask our presenters questions during the webinar. We will have time in the end, but please ask your questions when you think of them. You can use the same question field in your control panel to ask a question. We'll try to get to as many questions as possible during our time together. You'll receive an email tomorrow with a link to the webinar recording, and please see the handout section in the control panel for the handouts from today's section. There's also a short survey at the end of the webinar, only a few questions. Please take a moment and provide us with some feedback on our webinar today. We'll take advantage of any of those questions and anything that we don't get to during the time together. We'll make sure that those get addressed as well. So again, hello and welcome again to episode 9, this time featuring PatSnap. My name is Tony Trippi. I'm founder of ML4Patents.com and managing director of Pat Informatics. And I'm joined by Sam Wiley and Shiv Umesh Shankar, sorry about that, from PatSnap. So take it away, guys.
Thanks, Tony. So my name is Sam Wiley. Like Tony said, I'm the head of thought leadership and customer advocacy here at PatSnap. And today we're going to be talking about AI and ML, specifically for drug discovery, so the life sciences. And obviously, as we're going to get into later in the presentation, there's a huge overlap with the world of patent data. Patent data is extraordinarily important to this sort of work. So very quickly about me, my involvement with patent analytics goes back to my time at the USPTO in the early 2000s. Not too fun to say things like that, but I've been it's been my passion for at least the last 20 years. But especially maybe the last 10. So, I was involved with companies like Inography and others, and now here at PatSnap, and really excited about the idea of innovation intelligence influencing decision making in spaces like life sciences. So areas where you can very, very hand on your heart say that it's making a huge difference for the world. So very excited to be presenting today. Most of the talking today will be done by an actual expert in this area, not myself. So I'm being joined by Shiv Umesh Shankar, live from Singapore. So Shiv has been kind enough to stay up very, very late for us to present today. And Shiv is a data science product manager at PatSnap and an expert in this space. And Shiv, I'll let you introduce yourself.
Thanks, Sam. Thanks, Tony. Yeah. So I'm Shiv. I'm really happy to have this opportunity to talk about data science and how it's impacting patents, AI, ML, drug discovery, and development. So I've been with PatSnap for over 3.5 years now. I've had a decade of experience in bioinformatics and data science, specifically towards healthcare life sciences. Previously, in my role in PatSnap, I was a data scientist, so I was developing AI, ML algorithms that go into product development. In my current role as a data science product manager, what I do is I help customers understand their needs, help that transition into the latest state-of-the-art AI ML technologies that then go into benefit their customized workflows, both in drug discovery and drug development. So jumping on directly towards today's topic, so how does machine learning, AI ML, influence drug discovery and development, and what are we doing as an AI ML community and AI-led organization in PatSnap using the latest technologies powering through product development. So AI in drug discovery and drug development. So in today's talk, what I wanted to go through was show through what are the trends that are happening in drug development strategies, how much of an impact does AI have in life sciences and biotech in powering through their innovation. Where do we see pharma innovation happening in the next decade or so? And how does PatSnap use AI ML to support drug discovery and development using data state-of-the-art algorithms towards providing easy to use and convenient solutions? We have seen not just through the pandemic, but over the last decade, a phenomenal growth in biopharma industries. And this is not only just affecting larger enterprises, but also smaller biotech, right? So we are seeing growth witnessing across regions, across sectors. impacting small, big, and medium companies towards whatever form or shape they are involved in biotech and pharma. And this is across region, right? So you see growth in the US, you see growth in Europe, and you see a phenomenal growth in China compared to the rest of the world, almost doubling or tripling in size. Now, what's leading to this growth, right? So it's done by both innovation activities along with M&A, IPOs, and fundraising. And why do we see this happening? I mean, typically, as we see that we are going towards an aging population in the world, with an aging population, you have multiple other challenges coming in. You have chronic diseases, you have increases in life expectancy, you have a lot more older population, and with that, you have multiple other effects compounding, and along with that, you have pandemics arising with an increasingly overpopulated world, right? So there's always going to be a need for, and there's always going to be an impact on having the latest technologies coming through in life sciences and pharma. Given that there are opportunities for having a tremendous impact in life sciences and pharma, what are the main challenges that we are seeing? We do see that life sciences and pharma companies typically provide a lot of interest and investment in R&D activities. However, given that we are seeing an increasing spend in R&D, what we are also witnessing is that the costs are going up. This could be in the form of cost of producing drugs. It could also be because the average sales forecast for these drugs are coming down. What does this lead to is the return on investment on R&D is at an all-time low. What this means that the value of return for every dollar that's being invested in R&D is low, right? So how can we provide innovative solutions and latest technologies that give you more bang for the buck, basically in investing R&D returns? And typically, when you're looking at a pharma company, their portfolios encourage therapy area heads or data science teams to have diversified strategies. These can be through building up multiple assets. So these can be through multiple therapeutic modalities, investing in multiple avenues of research. So through new and improved mechanism of actions for that drugs, could be identifying new molecular targets for developing new drug development programs. It could also be at identifying newer diseases to target where there's growth in either more people or patients being affected, or there are newer diseases such as COVID-19 that come in and that require an urgent intervention in the form of newer drug development strategies from life sciences and pharma. So what this means is you're going to have an increasingly diversified portfolio in both in terms of R&D strategies and also business needs, right? So where do you put your money in? What's the best way to get that back? And where do you see growth happening both in terms of R&D? So where are drug development and drug discoveries programs happening?
Hey, Shiv?
And again, we are seeing this. Sorry, yeah?
Yep. We have a question before you start further into. One quick thing too, I don't know about everybody else, but on my end, you're breaking up just a tiny bit. And so
really?
It might be useful if you spoke just a little bit slower. I'm not sure if that will help with a little bit of a breakup, but maybe we'll be able to catch a few more words. I don't think we've missed anything thus far, but just something as we continue to move forward. Now the question, are there any good breakdowns? You're providing numbers as far as the size of the industry. Do you have any sort of an idea how much of that might be research versus development? And in this particular case, where you actually, you've been talking about pipelines, maybe you can address while you're talking through the pipeline a little bit about when it stops becoming research and when it starts becoming development.
Right. Sure. Thanks, Tony. So probably too small to see, but what we are saying is typically from conception of an idea to end product, right? So when we say end product, the drug being actually used in market, it takes around 10 to 12 years. So from idea generation, which is typically early in the stage, so that's around here. It goes through various phases in a drug discovery and development life cycle. So once the idea is generated, you then have in-house research being conducted in different companies or in academia. So this typically happens 2 to 3 years, right? Once we have a valid idea, valid potential for a new developmental product to take place, that then happens through another 2 to 3 years, providing preclinical research. So these could be trials in-house or in vitro or mouse models, so animal models, right? So by the time we have a solid concept, solid substance coming in, it's around 5 years from idea to having a substance that can go into clinical trials. Given that we have 5 years from idea generation to clinical trial validation, that's around 5 years. In terms of timelines in clinical trials itself, again, apart from taking the whole COVID experience completely, that's an outlier, right? We've had really fast development and really short terms of turnaround times in clinical trials happening. On average, clinical trials take anywhere between 5 to 7 years. And once we have at a late stage development, so when you say development, so 5 years in terms of discovery going into the clinical trials, and around 7 years from the whole development phase, from early stage clinical trials in phase 1 to phase 3 and more, that's a late stage trial, is around 5 to 7 years. So in terms of total timeline, this takes anywhere between 10 to 12 years for an average development life cycles for a drug. Hope that answers, Tony.
Sorry? Yeah, I mean,
would that answer your question? Yeah.
yes, I think so. Yeah, thank you.
Tony, it goes without saying, but pipeline's a bit of a misnomer, of course. It's more of a funnel. There's vastly more assets in discovery at any given moment than any stage down the line, all the way down to delivery, where there's obviously just a handful of valuable assets that make it through clinical trials.
Yeah, and of course that's the whole driver of the economics of the pharmaceutical industry. I worked for a couple of pharmaceutical companies at the beginning of my career, and it is really amazing. It's not just marketing hype. The number of compounds that even compounds that are in phase 1 that don't make it all the way and become an approved drug. So it really is brutal what happens as far as the economic model and making sure that the candidates are well vetted, prior to getting into these trials. So thank you.
Yeah. So again, going back to what we have had in terms of discussion, right? So the key ideas behind is, how do you reduce this 10, 12 years on time to market? How do we then reduce the risk of failure? I think average there are 1 or 2 drugs out of 1,000 which goes in from your idea conception stage to actual development in pharma and later on getting market access, right? And how do you use the latest technologies in AI that can help reduce the risk of failure and reduce the time to market? And this is where we want to focus on, and this is where a lot of time and investment has been happening. So biopharma companies typically want to change their shift in R&D model. So by using latest and most updated analytics and technologies, better partnering. So if you have earlier stage development happening through partnerships, activities across the world, then can I then use the same technology that's already been developed to a newer disease area, newer therapy area? Are there newer operating models, newer capabilities, skill set that I can identify using the latest technology or using the latest information that's coming out through patent documents? And could AI be the solution for this, right? So we've seen a lot of activity happening in AI in terms of investments, mergers and acquisitions, and also through drug discovery itself. So we have seen activity in the last couple of years where AI models have been used to identify lead compounds much earlier. So typically reducing the risk by identifying potential compounds much earlier than what would have taken through a traditional approach. Given that there are opportunities for growth in using AI in drug discovery and development, what are the main challenges? Challenges that I would foresee is in organizations want to use AI as much as they can to increase efficiency. It provides a value addition, and it also provides a way to reduce costs, right? But the challenge is how do you integrate AI into current workflows and processes? And how do you manage the associated risk, given that you have AI models running through a number of permutations and combinations? How do you ensure that the final generation of a candidate is well thought out, is well recognized? And how do you ensure that the cost of running through these billions of calculations doesn't overshoot your traditional expenditure? And how do you ensure that you have data that is needed to support your predictions and model interpretations, right? So that's where the challenge actually lies in. Now, in terms of barriers of using AI, apart from data and an provided state-of-the-art model costs, you also have a higher cost, initial cost coming in. So you need to ensure that you have costs coming in for providing access or getting access to the latest data that can then go into your model. You need to ensure you have the latest resources, be it in terms of computational resources or actual people looking into developing these models. So typically, AI model investment or AI investment in drug discovery and development happens on towards larger companies. So organizations with higher revenue invest more heavily in AI. What this does is you have your SMEs and small biotech kind of struggling to ensure that they have access to the latest technologies, the latest data that can then also boost through their drug developmental pipelines, identify or reduce risk in candidate selection, and so on. Again, given that we've been focusing a lot on life sciences and pharma, you do have adjacent industries coming in through bio and chem. Again, there's going to be a lot of overlap between what's being developed in pharma life sciences, whereas what it can transition into in adjacent industries in bio and chem. So when I say bio and chem adjacent industries, it could be agriculture, F&B, it could be consumer products, chemicals, and energy. A lot of development that happens is going to be on so DNA and RNA based technologies, recombinant DNA technologies, including biopharma development. It could be biomodeling, biomimicry, and biomolecules, right? And why I'd say there is a lot of overlap or potential to look into newer areas is, again, bottom line, it's still substances that would work in any different setup. So could I, given that I'm investing 10 years in this developmental strategy, could I then identify avenues where, you know what, I could spin off into a potential new partnership against an F&B company using a similar technology, but let's say for producing novel therapy mechanisms, on one hand, whereas could I use the same technology that has been developed for life sciences to improve food delivery or food processing, right? So what are avenues that I can tap to, given that I'm going to have a 10-year leak period to see potential returns?
You know, Shiv, I think I'd like to point out as well at this point that while this is a life sciences and pharmaceutical based and biopharmaceutical based presentation, you're absolutely right. And while that example you're giving is tangential to other verticals that are associated with that, there is a ton going on with, say, new batteries and material research, and you know, packaging materials, as you mentioned in the slide, but you know, those that don't have agricultural items, basically anything that involves either a biomolecule or a small molecule. These approaches are going to be relevant in order to do high throughput screening, in addition to modeling, to really cut down on the number of candidates and cut down on the development time, or the research time, I should say, in coming up with new candidates more quickly. So this is absolutely going to be applicable to all areas of chemistry and microbiology, not just life sciences and pharma.
Yeah. Thanks, Tony. Interesting, right? So going on, probably a decade earlier, I was working in academia as a research scientist. What I was doing was, again, I was scientist developing AI algorithms, but it was very similar. So the biomolecules that were involved in development could directly go into providing enhanced biomaterials, say for biofuel development, but the same technology or timelines could be applicable for, let's producing the latest therapy area. So could I use the same technologies that I've been developing towards producing a newer way of protein for influencing drug development, or could I use it for producing a newer technology for biofuels, right? So there's a lot of overlap, and the investment that's being done, it wouldn't go for a waste. It's just identifying smarter ways to ensure that you are transitioning into adjacent industries using the same technology that's being developed. What was interesting was in earlier time as a data scientist, as a data scientist in PatSnap, where I'm working on developing newer algorithms. What I was seeing is, let's say, for example, the enzymes that were used for providing the latest technologies for producing, let's say, in newer forms of plant-based food were very similar to the technologies they would use in antibody production. Again, they would use the same ways to, let's say, cure meat and so on, but it was sharing a high similarity. So there are ways that you can tap into the technology in newer forms.
Yeah, no, it's funny.
And
One of my jobs here at PatSnap is we have to sort of categorize companies, Tony, that we work with. And so when we're working with let's say someone developing a meatless tuna, you know, their technology is really a life sciences technology, but the customer they're selling to is people interested in meatless sushi. So it's interesting that the crossover.
again, so this could potentially, again, this what we are talking about was adjacent industries, but even within life sciences and pharma, there are sub-industries. So for example, medical devices, you would need to know a similar technology, a similar concepts behind medical device development towards your drug discovery or end product, right? How do you develop products? So there's a lot of overlap adjacency, and we do see this happening in patents too. So what this chart shows you is looking into analysis of patent documents that show how cross-domain applications function, right? So for example, what you see on your chart here on your y-axis is what are the typical applications that have been coming up via patenting strategies, and what are the companies involved. So you would see on your x-axis, these are companies, and you see that when they are trying to provide latest innovations, latest inventions or discoveries, there's always a cross-domain application happening. You would see, for example, there is always an involvement of glass-making apparatus in combination with drug compositions or medical preparations, right? None of the innovation or research that's happening is going to be focused solely on a single domain. There's always interdisciplinary activity happening, and this sort of provides the spark for finding out the latest innovations or discoveries, right? And this typically, the cross-domain applications, the industries that are coming in always talk to each other. So this is what we mean by when we say using connected data or connected innovations, right? So what we do at PatSnap, we are strongly believing that when you are at the forefront of innovation or trying to find the latest happenings and to inspire you in your R&D activities, it's always good to have this connected knowledge happening. So when I say connected data, connected life sciences data, what it means is you're connecting R&D activities happening across the world. It can be in the form of latest patents that are being coming up, latest scientific research happening, or what's happening in a clinical trial in China that could influence my R&D decision as a country in Scandinavia, right? It could be finding out latest technology. It could be finding out partners. So this is what we mean by connecting the world's disparate life sciences data. In order to do that, we use the latest in AI technologies. So when I say latest in AI technologies, this applies through both computer vision technologies and deep learning based NLP and AR technologies. So at a high level, it involves a lot of understanding of text that's being written in patent documents, that's being mentioned in scientific papers, clinical trials, regulatory approvals, and so on, along with what's captured or provided in the form of images, figures, and text. So that's image understanding. So what are the latest chemical structures that are being patented? What are the sequences that then tie into latest biologics, antibody therapies coming in? So a lot of investment and research in PatSnap goes in towards text understanding and image understanding in order to connect these dots to serve the community. So at PatSnap, how are we leveraging AI in our life sciences products? So we have our in life sciences product suite, we have Chem, Bio, and Synapse as our three main flagship products, each of them serving slightly different or adjacent industries, right? And how do we use this? So we use technology ranging from computer vision, machine learning, machine translation, in order and OCR, right, at different areas and different aspects. I'm going to be walking through some of the aspects and applications of how we are using these technologies in PatSnap's Life Sciences suite. And in order to connect these a variety of data. So for example, typically what we do is we're not restricted towards a specific angle. So we do ensure that whatever we use or apply AI through is across the drug development and drug compound life cycle. It includes articles mentioned in patents that are typically at the very early stage of drug discovery. So you're finding out ideas, concepts being mentioned in patents. How do you then connect that across the entire drug discovery and development lifecycle through news feeds, scientific literature, blogs, and who are the major players or sponsors behind these companies? And in order to do that, what we do is we used AI to come up with better features and capabilities that influence or improve the end user in order to provide insights, better insights, and better inspirations through enhance their current or existing workflows, right? So what we do is we use AI in a variety of ways in our solutions and products. So it could be through structure sequence highlighters, improved ontologies, enhanced landscapes and insights. So I'm going to be walking through each of these in the next few slides. Typically, how does data get processed through our life cycle, right? So when we say data, these are billions of data points. So we have a few hundred million patents in life sciences, pharma, or any associated industries. We also have scientific research documents, so scientific papers published all over the world, a few million of those, along with clinical trials, news, and regulatory approvals. But given that we have this vast amount of data, the key task and the key focus area is on content extraction. And how do we ensure that the content we extract can actually be linked through and be providing useful insights, right? So again, at the very start of our data processing is NLP. So it could be either your traditional CRF-based models, or it could be the latest deep learning models. And how and what we choose highly depends on the task at hand. It could be, and it also depends on the data that we are ingesting, right? So what we want to do is extract the raw data from patent PDFs. Could be extracting raw data from clinical trials. Once we've extracted these, how do we then classify them as sequences? How do we classify what computer recognizes an image? What type of an image is it? Is it a chemical structure? Is it a sequence, or is it just a chart that's showing up in a patent document? And once we do that, how do we then ensure that whatever chart sequence that has been extracted is actually the same that's being mentioned in patent documents across jurisdictions? So that then comes in the content normalization piece. We do ensure that we are able to relate to documents properly. I mean, there's going to be innovation happening across the world, happening in multiple different regions. So when there is innovation happening in multiple different regions, we do need to have the capability to ensure we are capturing whatever is the latest state-of-the-art technologies that's being built. So we do ensure that machine translation can capture in language agnostic research. So whatever it's being published in, wherever it's being published is, we need to extract that information out. We need to link it through and normalize it. So when I say normalize it, how do you ensure that, and for a typical example, a patent might mention NASH as a disease, another the patent or a scientific paper might mention it as non-alcoholic fatty liver disease. For an end user, they mean the same, right? How do we know that the NAFLD or NASH mentioning patent document can connect to a clinical trial that mentions non-alcoholic fatty liver disease? This is a challenge. And in order to ensure that we are successful in this, we need to build robust databases, right? And that's what we do. And we ensure that we are tapping into the latest technologies in AI and ML, but we also need to ensure that we do have a lot of content curation happening that feeds into ensuring that the models that are being trained on are training on the latest data, more comprehensive data, and the coverage is quite high.
Let me ask a quick question. Actually,
Sure.
quick questions. We got a question from the audience about trademarks. It's applicable to a degree, especially in the later stages, but maybe with other sorts of industries as well. Is there coverage of trademarks and the corresponding association with the companies through the named entity recognition? And I'm assuming named entity recognition in this case is actually assignee standardization. I'm thinking you actually mean it as named entity like NAH, the example you gave there. But that's probably one of the biggest things that I've found that's challenging in all of this is that corporate assignee or company name normalization across multiple data sources is bad enough when you got to do it with just patents. But then if you have to also bring in clinical trial data, if you also have to bring in journal literature, if you also have to bring in news feeds and financial data, and every turn of that crank just creates more and more difficulties when you're trying to associate the party of interest, as it were. So there were two questions there. One, trademarks, and then second, identifying the party of interest or that corporate source designation that's normalized throughout all these data sources.
Right. Yeah, thanks, Tony. So on the question of trademark, right? So if you're talking about it in the angle of life sciences and pharma, these could definitely be related to, let's say, your finalized or end drug products, so product names. Then yes, so we do ensure that we are capturing in latest technologies, both in terms of the end product, what is the drug or the substance being named as, and how can that be linked back to the earliest mention of the substance in a patent document, probably a decade ago, right? So these could be typically in a patent document, you wouldn't find names that are going to be the eventual product names, right? So these are trademark product. But what we do is we ensure that our AI technology can then relate to the document, the earliest patent document which had been published or applied a decade earlier to finalize the end product that happens in a decade later after regulatory approval, right? What we do is we have multiple ways to do that. We do tracking through multiple technologies too. So for example, how do we connect documents across a decade is we rely heavily on assignee or our company normalization, right? So and this is a challenge. So we have companies working across the world, so you have to take into account subsidiaries, you have to take into account partners. You'd also have to take into account M&A activities happening over time. So let's say a startup that might have initiated the concept right now might not exist 5 years down the line. So how do you ensure that the end product could actually be related back to the patent that was in the startup, right? So we have assignee normalizations happening over time. We do tracking changes over company names. And then what we also do is that is half the battle. But in life sciences and pharma, it's also important to know that you're able to track through time on how the actual product or the eventual product itself is being named. So we do ensure that we are, and again, so the biggest part is, I wouldn't say it's on extracting these entities, but it's on normalizing and linking them. So that's where the challenge is, right? So we do ensure that we are capturing those.
Okay, great. Thank you. Let's continue.
So an example as what we wanted to show you is how do we use the latest in computer vision, like OCR and NLP technology. So for example, a sequence mentioned in the patent document, if you're looking at older patent documents, could be mentioned in the form of images. So these are typically sequence listing files that can run into a few hundred pages. What we do using the latest technology is ensure that we can accurately extract out these DNA protein sequences and then relate it back into what they actually are meaning. So what is it used for? So it could be a single string. It's actually a sequence of letters, but it's important to ensure that the accuracy of extraction is very high. A single or a couple of changes in amino acids or nucleotide could entirely mean a new function that's being developed or discovered. And we don't want that. We want to ensure that whatever is being patented can actually reflect back into the application of it. So the challenges here is extracting out accurate and high quality sequences. Along with that, whenever we are talking about substances, biomolecules, sequences are half of it. The other major challenge is chemical structures, and we are developing our OCR technologies to enhance extractions of chemical images from patent documents, patent images, and text. Um, so NER normalization, right? So this is a challenge. So for example, a typical patent document might read through this. What we do is we are using our deep learning based models. So language models and language dependent and independent models could also be a CRF model, which is probably a dictionary based model, to ensure that when we are using the models on the AI for AI extraction on patent documents, be it through extracting them information from title abstract claims, how do you know that whatever is being written out, that a model can identify this particular word as a drug, disease, or gene, right? So for example, we have models that identify these entities, so identify these words in patent document as a drug, disease, or gene. Once we have that extracted out, we then use the associated sequence or chemical structure to link through the final eventual product that's being claimed in. So if a substance or sequence is saying, or sequence ID number 511 is actually the gene that has been developed by our R&D activity. So this gene could have multiple other impact and applications. So ensuring that we are linking different concepts in a patent document within each other, and then across linking these concepts across to the wider life sciences and pharma space is key and vital strategy that we are pursuing in PatSnap. For example, right, this is a real example taken out in terms of screenshots. So what we do is using our AI NER models. This was a clinical trial by a company. It was initiated in 2007. Around the same time, they also filed patent documents. So they had patent applications, multiple patent applications in the same time, right? So what we see is again, the colors here represent what our AI model is picking up. So what we do pick up is the disease terms, the drug itself, and also drugs, chemicals that might be mentioned in a clinical trial and in the sponsor's earliest patents. And why it's important to ensure that the normalization across happens not only in terms of company names, but also across biological entities, is that's how we are able to relate a document, a patent document, with a clinical trial. In this example, because we had both biological entity normalization and linking combined with company linking, we were able to identify and relate that a clinical trial that was initiated was actually linked to this earliest patent because both it contained the similar biological entities and had the same sponsor. We're able to also link to scientific activities happening a decade later in terms of scientific papers published on the disease or on the drug, in terms of company filings, and eventually sequences, right? So this was what was claimed in the earliest patent, but once we were able to extract out the sequences via images and text, the power of the linkage happens is you can actually then use the same sequence to look at FTO search, what's happening in terms of latest activity in the sequence space itself, or what's also happening in life sciences, pharma, or therapeutic area for osteoporosis. And what I wanted to highlight was over time, the same drug, which was abaloparatide in this example, goes from being called a research or company code, such as PA05A, to the finalized trademark end product, which was Timlos, right? So you have name changing for the same sequence over a decade. You also have companies tracking them through different forms, different variations, and across the world. So yeah, so for example, what I wanted to show in this image was this particular sequence, even though which was patented and filed by a company A, Radius Health, had very similar sequences in a number of other patents across the world, genuine jurisdiction. So we had sequences which were similar in WIPO, we had sequences similar in Japan, we had sequences similar in EPA patents, right? And why are we able to link these documents across? It's because we are linking both companies, we're linking the concepts in that and you're linking to the eventual end product, which might be a substance. So this is how we are tracking from early stage of concept discovery, concept initialization towards end product development. What we have been embarking on the last recently, last year and so, is using this connected data as a form of knowledge graph. So when I say knowledge graph, what we do is we have these billions of disparate data sets. How do we actually connect them in order to make meaning out of them? So we are using the latest in terms of knowledge graph technologies to ensure that there is a seamless connection happening across document types, across document formats, and across entity types by linking companies to biological entities. So that's how we are enabling our users to discover the latest insights and happenings in life sciences, pharma, bio, and care. And a core concept of it is machine translation. So and when I say machine translation, I mean what we are doing is we're using machine translation to understand and link documents across languages. In this example, what I wanted to show is alluding to what we were discussing probably a few minutes earlier, is how do we ensure that we are linking technologies and we are able to provide adjacent industries or adjacent we are able to provide insights that allow our users to move into adjacent industries given that they have a technology developed. So what's in common behind pest control, pest in plants towards reducing allergy in food products or and vaccine development, right? What was interesting was we were able to form a connection to all three of those by linking both the substances, biological concepts, and the eventual applications of those. So for example, the patents that are being mentioned here in US and China actually had very similar sequences that were being used for developing the latest technology used in terms of CRISPR-Time delivery systems, targeting towards allergy reduction or that could also have an application towards pharma development for vaccines, right? So we were able to identify the links through this in terms of connecting the dots by extracting the sequences out. We were also able to link them in by extracting the concepts out. So even though we were able to link sequences, we were able to link to documents via sequences. We are then able to link to a completely newer angle of application via extracting the technology or concepts out, right? So that's how we are using machine translation, both in terms of the actual substances that are being translated and transferred, and in terms of concepts. A huge factor of it is how do we organize these data, and in order to do that, how do we ensure that a document that mentions a substance A could actually be referred to in multiple other ways. So what we are doing is we're building out proprietary ontologies in life sciences and in companies and the technology. So for example, let's say how are drugs categorized, are diseases categorized, can a patent that mentions a very niche area of a disease A, could the technology developed there, could it then be having applications in a broader area? So for example, for a technology in development in a very small form of breast cancer, could it then have wider applications across all cancer therapies? Or could a technology such as CRISPR have wider applications, as we saw in the earlier slide, across different applications? Could be plant or food production to vaccine development. So this forms the meat of what we do, is linking biological entities with business entities, right? So when I say biological entities, these could be drugs, it could be targets, diseases, mutations across documents which contain both business entities and biological entities. So we're linking patents, scientific applications, regulatory approvals, clinical trials, in order to serve our clients who might be interested in order to find the latest research to perform drug repurposing, for performing competitive intelligence analysis, portfolio analysis, and so on. To summarize, what we want to do in our life sciences product suites is to connect the entire drug discovery and development life cycle. So these on your left are your biological entities. So it could be biomarkers, pathways, sequences, and here are your activities that typically are mentioned in the documents over the development lifecycle. So from the earliest patents to the latest news that mention the technology. So by connecting these over time, what we do is we are providing a platform, a single platform that helps the decision makers to get the latest insights in both drug discovery and development. So in order to aid the decision making strategies on where do they want to invest in, what is the latest technology happening? Do I want to focus on therapy area A versus B? So that's what we want to enable our end users and products through latest AI technologies. So in terms of product differentiators itself, right? How do we ensure that our products meet the needs and serve the needs of our customers? Even though we have all these complex data and complex processing happening in the backend, it's vital that the end user focuses mainly on the insights that is being generated. So ease of use is one of our key focus areas and priorities, along with combining the latest in AI-powered data curation, knowledge graph to link these different documents, companies, and biological entities together. Whatever said in whatever research or whatever search activities that are performed, it's easiest to interpret in the form of charts. So providing custom chat dashboards and charts, integrating diverse data sources right from the earliest in patents to the latest news activities, and ensuring in a highly connected and global world, we want to ensure that participants or people across the world have access to research happening across the world. So given that PatSnap has a wide presence in both East and West regions, we are providing a single platform that can connect East and West life sciences data in one platform. Example of screenshots. So I would think if time permits, I would actually walk through a couple of examples in the call later on in the actual product itself. So this is a knowledge graph for repurposing. Sorry?
Yeah, time check. We're at 10 minutes to the top of the hour. So we probably need to move quickly into the product. If you're going to move into the product, there's 5 questions waiting as well. So,
sure.
We're running very, very short on time. And so it's probably a good idea to keep moving and get into the product.
Thanks, Tony. Yeah. So I'll walk you through the knowledge graph in the product itself, and probably a couple of others. So again, as mentioned, even though this is a drug, we are actually linking it to the sequences, to the approval activities. Chatbots and visualizations are key. In this example, what I wanted to showcase was how do you use insights across different documents, across different areas. So how do you use patents, filing trends to proactively identify shifts in portfolio strategies? What are the developmental clinical pipeline changes that influence developmental activities? How good or how important is the target based on the activities of that mention a target in patent scientific research papers and so on? Can I also do a simple search to find out signals apart from noise? So how can I use or search in the latest technology to see what's happening? Who are the major companies working on? What are they using this technology for? So these are key use cases and aspects that our product serves and needs to. So quickly share the demo. Right. Again, so I had pre-run the search in order to save time. So what I'm showing you here is one of our platforms called Synapse. It's in a beta stage right now. What I had done was search on a disease called ulcerative colitis. What you can see here is it identifies and mentions the disease, ulcerative colitis, across different document sets, papers, patents, clinical trials, and news. And if I wanted to see what's the latest happening, right? So across all the news that has been mentioning ulcerative colitis disease, who are the top companies working on, what are they using this disease for? So I can then extract out information from this and use that in a knowledge graph. So populated the knowledge graph here. So what this tells me is using the information that was extracted out, it actually forms both a biological connection and a business connection to it, right? So there are these X number of diseases. So 3 red dots are actually drugs that are focusing on a disease here. identified in blue using the target here. And why is this key? Because we are merging both the activities that are conducted in business, so companies filing patents, companies filing latest clinical trials and development, and how does this impact the end biology to it? How does it impact actually drug development and discovery? And once you find that activity, so you can then, look into a specific company. So this was this drug identified in our knowledge graph by Gilead. So what is Gilead actually working on? So we do inform our users by looking into not just their current research, but what's happening over the last decade. So what are they investing in, and who are they working along with? And in terms of charts and timelines, so on a specific disease, can you create landscapes as to, can I identify partners to collaborate with? Can I identify the latest activity in patents, drug development, and diseases? Let me stop now and quickly go back onto the presentation, which I think is almost done.
Yeah, we've got several questions, so let's go ahead and get to those as
Let's
quickly as
go
possible.
to the questions. Yeah.
Okay, so
Yeah.
through the questions. This one was really interesting. It's my understanding that it's generally very difficult to link specific R&D activities to inventive output, such as a patent. Do you use proprietary data from companies to link the different stages of drug development pipelines? So you alluded to this, but are there specific sources of drug development pipeline data that you can then connect to the corresponding patents? I mean, I know you've mentioned that, but I wanted to make sure that this question was answered specifically.
Sure. In terms of specific data, yes. So we do have access to conferences. So let's say whatever the latest research is happening on that, these are typically published in an earlier stage in conferences. So we do track right from conferences, early stage theses or patents to end goal. But in terms of how do we manage to link these across, it's through our AI technology, entity linking and normalization. That's how we link them across time.
Okay, very good. Another one along the same lines, do you have an estimate, even a rough one, of what share of drug patents end up going into a clinical trial?
I would say less than 5 to 10%, Tony. Just a number off top of my head and going by whatever research is happening. You do have 5 to 10% of patents. When I say 5 to 10%, this includes evergreening of patent, includes various forms that eventual product might come out. But how do you relate it back to the actual concept? That's around 5 to 10%. The rest of it is, again, it wouldn't say it's irrelevant. That's what is spurring on to the end goal, right? So the technologies that are being patented in actually results in the drug development, but actually 5 or 10% is what directly relates to the end product.
All right, so another question that came up had to do with the development of these machine learning algorithms. How much of this material is developed in-house, proprietary, versus how much of it is open source?
Sure. Again, so what we did was when we started out this probably 3-4 years ago, we were trying to say, you know what, this open source model seems to have the best state-of-the-art results. Can we then directly transition into a product development? Unfortunately, it only serves to a certain limit of accuracy, right? There are complexities, as you know, in patent documents and clinical trials. There is no direct translation of an open source model into an application in AI or drug discovery life sciences. What we do is we enhance it over time using our in-house curation and using our in-house expertise in machine learning and AI to enhance whatever has been the state of the art in open source. But then how do you apply that to various complexities that are associated with patent documents? So, yeah.
Okay. And then final question, I think I have one that I'd like to ask as well, but we had a question about how the application domains are selected. So clearly you're not taking every single patent and putting it through all of these types of mechanisms. You're not manipulating or doing entity extraction or working with every patent with this entire workflow. So what do you do to identify those documents that you're going to put into this more rigorous life science approach?
So we take into multiple angles, right? I mean, one is how the patent classification itself is structured, so your IPC, CPC. But given that, you would see that it's heavily dependent on the patent examiner and how the patent itself is structured. But we do have one of that is IPC, CPC. Because you also have the organization behind and multiple other angles that, again, putting in a few million documents to the entire NER workflow is going to be computationally intensive. But given that we have these metrics that can help us identify the right set of documents, in a combination, it seems to help us identify the key documents, taking into account both the companies who are working on it, what are the actual technologies, and we have multiple avenues, right? We're not only looking at a single patent, but we're also extracting structures and sequences. So we do tackle them in a multi-pronged approach.
Okay, so and this will be the last question, and we'll go ahead and wrap up. Please take the survey at the end. Please provide us with some feedback. I think a lot of companies have struggled with the idea of working with universities and the idea of academic research into these various areas. And a lot of universities are beginning to come up with their own drug development programs. And ultimately, a lot of them end up passing it off after it gets past, say, preclinical onto a pharmaceutical partner, but they're still interested in taking on some of these early stage items themselves. Are there any particular additional challenges, or what have you found with regards to being able to run through this workflow when you're talking about organizations that are academically based?
Sam probably could chip in later on, but given what we've experienced, what we have been talking to in terms of academic partners, is they have a lot of technologies in terms of technology development, almost in terms of speed, equal to what's happening in your industries in pharma, life sciences. But the challenge they see is how do you then commercialize this, right? So we have academic organizations coming to us actively, mentioning to us they have this technology, they have this patent, but then they want to identify partners that can then take it to the eventual commercialization angle of it. So they use our platforms to identify partners to collaborate against, partners to talk to, or even research activities that then they can then perform in combination.
Yeah, Tony, and another angle we've been seeing a lot from our, I guess this is more from the corporate side, is an interest in finding the right academic partner. I think there's a desire to go outside of whatever you in your particular field see as kind of the top 5, top 10 academic institutions. I think those are, again, they have their own programs. They may have exclusive partnerships. So there's a lot of interest in finding, hey, I know there's a lot of good research going on at state schools in the United States, or even schools that you may not be aware of in Africa or India or other places that you just haven't done any business with before. That's where we're seeing a lot of interest from our client base, is how do I find the researchers? How do I find the research going at those institutions that aren't the really obvious ones that maybe I've partnered with in the past?
All right, well, we are at the top of the hour, and so we're going to go ahead and wrap this up. Please take the survey, and I just want to say thank you. Appreciate your time today. Fascinating. Got a couple of comments that have already come in about how really phenomenal it is to be able to grab all these different data sources and be able to look across them and have them work together so seamlessly. And so congratulations and best wishes with the product as you continue to bring it forward. And thank you everyone who attended today. And again, you'll get a link with the reporting. And if there's any other questions or comments, please go ahead and send them to me. There'll be my email addresses in the materials you receive tomorrow. So with that, I'm going to go ahead and close the session. And again, thank you so much, and have a great remainder of the day. Bye now.
Thanks, everybody.
Yeah. Okay.